So let's further explore the concept of dominance. And because we understand things on a molecular level already, because we understand how genes express proteins and proteins interact with each other, we can take this a little bit further. Now, complete dominance is what we've talked about so far, this idea that when we have a phenotype and that is a result of a heterozygote, the dominant allele will express itself. Okay. Now, you can also have situations where you have what's called incomplete dominance. And this time you have a heterozygote that has a phenotype that is an intermediate between the two alleles. So you don't have a dominant recessive allele, you have incomplete dominant alleles. The final relationship that we can see as a variation of this theme of dominance is called codominance. When instead of getting an intermediate phenotype, you get both phenotypes. So both alleles are expressed distinguishably. So let's look at an example of that. So incomplete dominance, you take snapdragons instead of pea plants, and you have red snapdragons and you have white snapdragons. And so we're not going to write the genes as uppercase, lowercase, because neither one is either dominant or recessive. So we use a different kind of nomenclature. We use a superscript R for the color locus, red, and a superscript W for the color locus, white. And when you get an organism that is heterozygous, CR-CW, you get a pink flower. So everything plays out just like it did before. Half the gametes from the heterozygote will be CR and half the gametes will be CW. Were you to do a self-cross here, then you get a ratio of phenotypes that is exactly the same as the ratio for genotypes, because every genotype in this case now is distinguishable on a phenotypic level. So if you're CR-CR, you're red. If you're CR-CW, heterozygous, you're pink. And if you're CW-CW, then homozygous white, then you're white. All right. So when we see this, and you can see how this could play into the idea of the blending hypothesis, but in fact it is still discrete units that we know as genes that are contributing to this. So incomplete dominance is another thing to consider when we look at the variability of genetics. Now, the other thing to think about is codominance. So codominance is when you have both alleles expressed on a phenotypic level. So the best example here is blood type. So the main blood type A, B, or O is dictated by a protein on the surface of the red blood cells. So in the case of a phenotype A blood type, it means that you have the A protein. And if you're B, you have the B protein. Well, you can also be AB. So that means you have both proteins. So you can see the triangles and the little circles on the surface of that cell. This now is a case of codominance. Both are expressed on a phenotypic level. Right, we'll come back to this example in a couple minutes when we start to talk about multiple alleles, but let's hold off on that for now. So again, the relationship between dominance and phenotype. So the alleles are the alleles. So the genes are the genes, and the proteins they produce are the proteins they produce. They're not necessarily battling it out on a cellular level. It doesn't make any sense, right? So all an allele is a different version. So recessiveness or dominance really depends upon what level that you're considering the gene expression. And we'll use Tay-Sachs disease as an example of this. So it's a fatal disease. The mutation is a dysfunctional enzyme that causes accumulation of lipids in the brain. So on an organismal level, the allele is recessive. So i.e., you would not develop visible signs of this disease on an organismal level if you're a heterozygote, because the healthy version of the gene is going to dominate its dominance, right? So the diseased gene would be recessive. On a biochemical level, the phenotype would be incompletely dominant, right? It means that your enzyme activity of that particular cell is going to be an intermediate. So intermediate between the normal level, if you had a homozygous normal version, and homozygous recessive, meaning you have very, very low enzyme level. So on a biochemical level, the phenotype is incompletely dominant. On a molecular level, it's going to be codominant. And on a molecular level, it's always going to be codominant. because both genes are expressed, both proteins are present, and then the activity leads then to cellular and organismal effects. But if you're just talking about the molecules being there or not, the alleles are codominant. Does that make sense? It's something to think about. So again, how does this work? The genetic deficiencies take out a lysosome enzyme. It's responsible for breaking down the fat. So we can look at these mechanisms and again consider them on an organismal, kind of a cellular or a molecular level. Now, a common misconception when it comes to dominance is that dominance, if a gene is dominant, it will automatically become more abundant in a population. And that is absolutely not the case. So the only thing that dictates whether an allele becomes more dominant or not is evolution, and that means that it is selected for. Selective pressures say that that allele will only become more abundant if it results in increased reproductive advantage. And that's a concept that we'll talk more about as we talk about evolution. But a very poignant example is that polydactyly, or the trait of being born with more than one or an extra digit, is dominant. This is a dominant allele. And I don't know how many people you know that are polydactyl. I knew one person in my life that was polydactyl. And so this is clearly, we don't see more and more people with 6 fingers. This is clearly a dominant trait that is not overwhelming the population. And again, that comes away from the genetics and goes into evolution. Now, the last thing that we want to consider when we come to this kind of variety of dominance is multiple alleles. Everything we've talked about so far has been either yes or no, purple or white, right? The gene for white or the gene for red in the snapdragons, right? So in some cases, you actually may have more than just 2 alleles. So now you have to remember you only have 2 slots, right? Because you're diploid. So you can only have 2 versions at any given time, or if you're homozygous, 1 version. You'd never have all 3 at the same time. But the 3 different versions are out there in the population. So if we go back to blood type as an example of this, we actually have 3 different alleles. And so we're going to use the I symbol, capital I, for idiotype, that is going to represent this locus. And so I superscript A is dominant A, and I superscript B is dominant B, and little I then represents the recessive version of the gene, which is O phenotypically. So A and B are both superscripted, so they have a codominant relationship, but I to either one of those is recessive. So again, when you look at that, you have 4 different combinations that you can get. You could either be, from a phenotypic point of view, you can either be A, B, or O. From a genotype then, if you're A, you could be homozygous A, or you could be A little I, and still have the same phenotype. If you're B, it's the same thing. You could be homozygous B or heterozygous B little I. If you're AB, you have the codominant relationship AB in your genotype, and the only way to get O then is the homozygous recessive little I, little I. All right, so think about that. The key to that idea of multiple alleles is that the relationship between each separate allele needs to be determined. Right? So in this case, A and B are codominant, but you could have 3 alleles and they may not be codominant. You may have one that is dominant to all the others, another that may be dominant that is recessive to one, but not recessive to, but dominant to others. So these have to be taken on a case-by-case basis.